In this experiment, I looked at the phenomenon known as complementation using various strains of yeast that have a mutation in the ADE pathway that causes them to be pink. Complementation is a phenomenon by which the effects of two different non-allelic mutations in a gene are partly or entirely canceled out when they occur together. Complementation test, also called cis-trans test, is a test for determining whether two mutations associated with a specific phenotype represent two different forms of a mutation, are allelic, or are variations of two different genes. In this experiment, a cross will produce a white colony if they are complementary because the progeny will be heterozygous for the mutant allele at each colony and therefore cancel out the mutant red phenotype. For this experiment, the organism that we will be testing for complementation are yeast cells. While asexual reproduction is most common in yeast cells, we will be experimenting by crossing over two haploid spores that have a mutation in the ADE pathway. Sexual reproduction in yeast involves the joining of a haploid alpha cell with a haploid A mating cell. When an alpha and A cell are physically close, they are able to fuse and form a diploid zygote. This zygote is able to then asexually divide and form a genetically identical population of yeast cells. This is particularly helpful in a complementation test because it allows for the identification of the mutations present in the parent colonies based on the color of the filial colony. The ADE genes are responsible for the biosynthesis of AMP, which is adenosine monophosphate. The yeast that we are studying all have a mutant form of the ADE genes and are therefore unable to biosynthesize adenine on their own without one of the precursor molecules in their growth medium. This particular precursor molecule is known as AIR, which is responsible for the pink phenotype. The mutation in yeast cells that turn red must be located after this AIR pathway. The pink color is due to the buildup of AR molecules, which are then subsequently oxidized to form the pink or red color. If there is an excess of adenine in the growth medium, then the yeast will no longer be pink due to the cell already having abundance of adenine that is ready. It will no longer need to spend the energy to produce it the long way with precursor molecules. My hypothesis is that as long as the inoculating loop techniques are adequately done and sexual reproduction takes place in all of my dots, then two of the dots will retain the pink phenotype and allow for successful identification of the three strains that contain the same mutation. Because if the two strains are non-allelic, the colony produced will be white, but if the strains are allelic and complementation does not occur, then they will remain pink. Therefore, I think that the majority of the colonies will exhibit white phenotypes and show complementation. The methods used in this experiment involved crossing 6 different strains of yeast, all exhibiting a red phenotype. In order to do this, I used an inoculating loop and sterile technique and placed them in a 3x3 grid for a total of 9 crosses. The top row going down were the 3 alpha strains, and the side row going across contained the 3 A strains. The alpha strains for my plate were A2, C1, E2, and my A strains were B4, D1, and F2. After the colonies were placed on top of each other in order to promote sexual reproduction, they were then allowed to incubate for a week before analysis. The results were then analyzed based on the color of the colony produced. White phenotype meant that complementation had occurred, and red meant that the two parent spores had the same mutations. My results were lucky in the respect that none of my colonies appeared to move during the incubation period and were able to stay in the same spot in order for more accurate interpretation of my data. For my individual crosses, it appears that definitively a mating yeast sample B was complementary to all 3 alpha mating cells. Likewise, the alpha mating cell C appears complementary to all of the A mating cells. One of the ambiguous results that appears that it could be either pink or white is the cross of alpha E and A mating D. While there appears to be a pink center, there also appears to be a white rim surrounding the colony. Therefore, I declared that complementation had occurred. Otherwise, there would be no white at all in the colony. For my results, it seems plausible that A, D, and F all had the same mutations because they retained a pink color almost exclusively. These are the only 3 that I can accurately presume to contain the same mutations because they were the only ones that maintained the pink phenotype. The map shows that when mutations F, A, or D are crossed with the other 3, it will produce a white colony. But when F, A, and D are crossed with each other, then they will maintain the pink phenotype. The next step in determining whether my results are viable would be to cross alpha A with the A mating colonies D and F to ensure that they still remain pink and prove that no complementation had occurred. Based on the map, there are 2 possible errors. Either I made a mistake when placing the colonies and double placed one of the 6, or there were more than 2 mutations present in the samples which allowed for a greater number of complementations. I learned many things from this experiment. I now understand why yeast is such a good tool for understanding genetics and in the laboratory and how they reproduce. I have a better understanding of what complementation really means, as well as how to identify if it has occurred. I also gain an opportunity to better refine my sterilization techniques and use of the inoculating loop, and I'm happy with the results on my plate.